nucleus finite rate is proportional to the amount of mismatch. So the structure that I showed you before, the more the mismatch exists between bottom-up input and top-down expectation, the higher the finite rate of the nucleus will be. So there is a measure of how wrong the cortical expectation was. Finally, higher order thalamic areas basically go through the same mechanistic stage with the difference that the input to the higher order cortices is not a sensory anymore, but is a complex output of a previous cortical stage. And this is an example of how the model works. So let's imagine we are given a bottom-up input, which is an horizontal bar, and the top-down expectation is also an horizontal bar. So this expectation, let's say, is that it's generated by another cortical area, travels down the circuit, and then hits layer 6. And layer 6 basically sends top-down feedback to the thalamus. So the level of the thalamus, this feedback has two effects. As an effect of direct excitation of the consistent set of cells that is appropriate for that particular stimulus. So it thinks or it hopes to see an horizontal bar, for instance, if that was what the neuron was trained to see. And also it has a broad of surround, so this is generated by a set of inhibitor interneurons which wrap the thalamus in this nucleus called the thalamic reticular nucleus. So one of the prerequisites for neurons of the thalamic neurons to fire is that they have to receive at least at least one source of excitation. So in this case, they receive bottom-up inputs, but they also receive top-down excitation, which is balanced by inhibition. So the neurons which receive both sources of excitation can fire, whereas anything else in the inhibitory surround cannot fire. And this is particularly important in the case of mismatch. So let's imagine that the neuron is expecting an horizontal bar, whereas a vertical bar is presented. So only the neurons which receive both bottom-up stimulation from the sensory periphery and top-down expectation from the cerebral cortex receive two sources of excitation versus one source of inhibition. So therefore the neurons in the thalamus can fire and propagate their activity upstream to the cerebral cortex, whereas neurons that are mismatching, they do only receive excitation from the bottom-up input, but they also receive inhibition from the expectation without the support of the top-down expectation, and that causes a reduction of activation in the thalamic nucleus. This reduction in activation in the thalamic nucleus is then sensed by the non-specific thalamic nucleus, which then projects to the superficial layer of the cerebral cortex, where it triggers a series of events that I will summarize very briefly here. So the non-specific nucleus projects to the apical, to the superficial cortex, superficial layer of the cerebral cortex, where its activation is picked up by a wide array of layer 5 pyramidal cells. And these layer 5 cells, through projection through layer 6, they basically fire synchronously and cause a reset in the cortical area by shutting down temporarily layer 4 cells. What this causes is basically a reset of the active conjugative code in layer 4. So we can see here a simulation example in which a layer 4 cell was spiking expecting a particular stimulus, which was then mismatched, and the reset basically causes that particular layer 4 cell to shut down and another cell to start firing and start to learn the new mismatched stimulation. So there is a lot of evidence that suggests that the non-specific thalamic nucleus is involved in the early detection of mismatched information. And this is particularly true in the so-called mismatch negativity literature, which is basically a brain event potential or brain wave, which occurs whenever a novel stimulation is presented which is embedded in a stream of homogeneous stimulation. So if, for instance, you take a guinea pig and you give a sequence of homogeneous stimuli and then present a stimulus with a high intensity, you will basically cause a mismatch negativity wave, and experiment recording in the brain has been showing that this particular wave is generated in the non-specific thalamus. And the basic mechanistic hypothesis is the following. So as you present the stimulus, of course the animal is not expecting the stimulus, but as you repeat the stimulation, the animal is going to build up expectation, more and more precise about the stimulation. So as you go through time, there will be more and more matching and more reduction of the activation of the non-specific thalamic nucleus, since this nucleus is sensitive to the amount of mismatch. But whenever you present a new stimulation, for instance, changing the intensity of the sound, then the nucleus will start to fire to capture this mismatch. And that's exactly what the data show, and we prove that our model qualitatively match this data. Also, the model tries to make an attempt to predict what happens when you start to stack various of these models together, if you're building, for instance, a larger cortical network. So let's imagine that you are basically, reading a book. Let's imagine that you're reading a book while listening to a melody, and somebody plays a trick and changes one of the notes, or start to change a few of the words of a song that is familiar to you. This will basically cause the particular brain modality, which is analyzing the mismatching stimulation, to start firing. In particular, the non-specific thalamic nucleus will start firing. And we also know that the non-specific thalamic nuclei are organized in a mutually inhibitory network, in the sense that whenever a non-specific thalamic nucleus fires a lot, it tends to inhibit other non-specific thalamic nuclei. And this is basically a mechanism, and there are some simulations that I will keep presenting you, through which a particular modality can start to shut down other modality by subtracting one of the main source of excitation to the cerebral cortex by mismatched based processing. So an area that experiences something wrong can rapidly drag attentional resources without knowing in advance where is the mismatch or where the potential wrong information is. So this is a very interesting and possible useful mechanism in a large distributed network. So we used spike time independent plasticity, which is a biologically motivated learning law, to learn oriented filters, because our stimuli were basically some very simple oriented bars. We tried to keep at least one thing simple, which was the stimulation. So we learned bottom-up filters and top-down expectation, which were shaped as the input, and that was one of the basic idea of the model. But also we looked into other things, such as what is the effect of top-down stimulation on the target population. So if you are expecting something, what happens to the neural field where the expectation is generated? And one of the things that has been observed physiologically is that top-down feedback has a modulatory effect on the target population. So this is a recording in the thalamus where you can see a little blip of activation, which is not enough to cause a spike in the receiving population, which is then followed by a longer lasting and deeper inhibition. And this is exactly what also we find in our model. And we can explain this by what is the architecture of the feedback. So as I showed you, the feedback goes down to the thalamus, but also aside from exciting the target excitatory cells in the thalamus, also excited a broader surround of inhibitory cells, such that the effect of direct excitation is compensated by a broader of surround. This is not only helping to calculate the match in the correct way, as I showed you before, but also allows the system not to continuously hallucinate or self-confirm its expectation in a way that is obviously not adaptive. And this is a recording of the model. The top plot shows the membrane potential of layer 6 in the cortex as it generates a spike and sends down its excitation down to the thalamus. So you will see here the little blip, which is the spike of the center in the middle, and you will start to see here excitation developing, consistent with an horizontal bar, which is then followed by inhibition. Another thing that we notice in the model